because I have her voice.
So, yeah, I want to introduce her. It's a pleasure to introduce Dr. Margaret Livingstone. He's a professor of neurobiology at Harvard Medical School with a keen interest in the ways in which vision science can understand and inform the world of visual art. She is the author of the groundbreaking book Vision and Art: The Biology of Seeing, which demonstrates how the cellular structure of our eyes and brain determines how we see and process artworks. She received her Bachelor of Science from MIT and got her PhD of neurobiology at Harvard University. In 1983, she became an assistant professor at Harvard Medical School, and in 1988, she was promoted to professor. She has been involved in studying numerous aspects of neurobiology, including visual and facial processing and subjects with dyslexia, which impact the field of studying learning disabilities. She studied face recognition processing in monkeys. She used MRI technology to localize the function of neurons within the temporal lobe, and this has significant implications for a study of autism. She was elected as a member of the American Academy of Arts and Sciences in 2015 and the National Academy of Sciences in 2020. I am very pleased to present to you Dr. Margaret Livingstone. Dr. Livingstone, the stage is yours.
All right. And you can hear me now.
Yes.
Good. Perfect. All the way across the world. That's kind of impressive. Okay, I want to talk about what art can tell us about the brain. People often say that I try to reveal things about art, but it's actually the reverse. I try to reveal things about the brain because of what artists do. So I'm a visual neurophysiologist. I try to figure out how we see by recording from neurons throughout the visual system, but artists also try to figure out how we see, and many artists do experiments. So I'm going to talk about works of art. I mean, I know there are works of art that are important because they reveal things about human culture or human society, but I work on low-level vision, so I want to talk about works of art that reveal things about how we see. So for a very long time, artists have used line drawings to portray their world. And it's so intuitive and so universally accepted that a line can represent a contour in the world that you have to step back and think for a minute that there aren't lines out there in the world. What there are is contours and discontinuities, and artists use a line to represent a contour or a discontinuity. And the reason this works so well is because of the way your visual system processes the pattern of light that comes into your eye. So Steve Kufler discovered that if you shine a spot of light on the retina, you get a barrage of signaling from retinal ganglion cells. But to his surprise, when he used a bigger spot of light, he got a worse response. And he figured out that that's because retinal ganglion cells are activated by light in one part of the visual field, but they're inhibited by light in the immediate surround. And so he found out if he shined an annulus of light on the retina, he got a suppression of firing. We call this center surround antagonism. Young people call it normalization, but it's basically the same thing. Retinal ganglion cells are excited by light in one part of the visual field, but inhibited by light in the immediate surround. And this is the most fundamental computation that your visual system performs on the pattern of light that hits your retina. And in case you don't believe me that this computation is ubiquitous, I hope you see little gray spots at the intersections of these white lines everywhere where you're not looking. And don't worry about why it's where you're not looking. That's just retinotopy or magnification. It's postgraduate. But the reason you see these little gray spots is because center surround cells at the intersection have more white in their inhibitory surrounds than center surround cells in the cross pieces. So they're less active. So you see a little gray spot. So if you take away anything from what I'm talking to you about today, I hope you take away the idea that vision is information processing, not image transmission. It doesn't do you any good to send a high resolution image to the back of your brain because there is nobody up there to look at an image. What you send is information in the form of neurons signaling. And these computations have to be simple because neurons just fire or don't fire, right? It has to be local because neurons are cells. They're little tiny things. And it's off an opponent. Neurons can either excite each other or inhibit each other. That's all. So if you have an image like this, that's light on one side, dark on the other, center surround cells over here are inhibited, center surround cells over here are not active. It's only center surround cells at the discontinuity, at the intersection, where there's an imbalance between the center and the surround, where you get a signal. So center surround cells automatically turn a contour into a line drawing. And this is information processing. It's image compression. If you were to save this image as a TIFF file, it would be huge, right? But if you saved it as a JPEG, which is an image compression program, it would be a much smaller file. And that's because JPEG, like your visual system, tells you where there are discontinuities and doesn't bother sending any information to you about where nothing is happening because there's no information there. People often draw center surround cells as a difference of Gaussians, with the cells excited by light in a small part of the visual field and inhibited by a larger, broader, shallower surround. And after images is what happens when you have two sets of neurons in opponency and one of them gets tired because it runs out of transmitter or something. And so you get an after image. And what you see in an after image can tell you what's going on in opponency. Our meeting has been upgraded. I have no idea what that means. You can infer the opponent computation that's going on in your brain by what you see in an after image. So as I told you, you have center surround opponency and luminance. And so if you look at a white spot on a gray background, even for just a few seconds, you'll get a dark after image. If you look at a dark spot, again, just for a few seconds, you get a light after image. If you look at a red spot, I hope you'll get a cyan after image, which tells you that you have neurons that are excited by red and opposed by cyan. If you look at an inward moving spiral, you will get an outward moving after image, which tells you that inward and outward motion act in opponency in your brain. So the visual world, the visual scene, is mapped onto your retinas because of optics, and this retinotopy or mapping is transmitted to early stages of visual cortex. So nearby neurons in early visual cortex encode nearby parts of the visual field. In your brain, all long-range connections are excitatory. Well, in your cortex, all long-range connections are excitatory. Inhibitory neurons are always local, short-range. They don't go very far. So that means that this opponency, this surround opponency, which requires this extra synapse or inhibitory synapse, has to be local in space. And you can see that in this illusion. All these dots, all these disks are identical, and I know they are. They don't look identical to me either. I know they're identical because I made this. What's graded is the surround. So opponency is local in space. So the dots that are against a dark background look lighter, and those that are against a light background look darker. You can see this in any work of art that is supposedly illuminated by a local light source. So in this painting by Terbruygen, the gist of the painting is that this is the sole source of illumination in the scene. But this is physically implausible because there's more light coming to your eye from this boy's shirt than there is coming from the supposed source of light that's illuminating the shirt, namely the candle. I took a photograph of my son blowing on a candle with a little pipe thing, and the photograph relays the veridical information about the luminance in the actual scene. And of course, light falls off with the square of the distance from the source of light. But I was there, and it didn't look like this. It looked more like this, in that I could see that my son had on a light colored shirt, and the shirt did seem lighter than the source of the illumination, namely the candle. This happened because I was filtering this entire scene through my center surround cells, which gave me information about local contrast, not the actual level of illumination anywhere in the scene. And of course, artists take advantage of this. The fact that our eyes detect only local contrasts allows artists and photographers to compensate for the limited range of lightnesses you can achieve on a flat surface. So the 3D world can exhibit a huge range of lightnesses, many orders of magnitude, but a flat surface like a canvas or a piece of paper can only achieve around a 20-fold range of lightnesses. That is, the whitest white pigment reflects only 20 times as much light as the blackest black. But by manipulating the background lightness, or by counter shading, artists and photographers can achieve a much larger apparent range of lightnesses in their works of art. So contrast is also locally opponent. These checks are identical to these, but these look low contrast because they're surrounded by high. These look high because they're surrounded by low. So contrast is locally opponent. Orientation is locally opponent. The center disk stripes are vertical, but they look off vertical because they're surrounded by another orientation. Motion is locally opponent. I assure you that red dot is moving vertically, but it doesn't look like it is because of the motion in the surround. Color is locally opponent. Each of these rings is the same identical gray. I know it is because I made this, but these look more blue, that is less yellow, because of the yellow background, and these look more yellow, that is less blue, because of the bluish background. Here's an illusion based on that. I need you to look at the center dot for just a few seconds, and I want you to keep in mind that what you're looking at, or what you're seeing, is white letters. And I hope you'll be a little surprised to get a yellow letter after image from white letters. But of course, that's because of the surround opponency in color. Of course, artists know this. Monet painted a bluish shadow with yellow highlights. So vision, I hope I've showed you in many ways that visual computations, at least early in the visual cortex, are simple, local, and opponent. But this is also true for even higher level visual computations, such as the ability to see distance and depth. So you look out on the visual world and it looks three-dimensional. Now, I know the world is three-dimensional, but the fact that you see it as three-dimensional is computed by your brain starting with two flat retinal images. That's all you get to start with. It's two flat retinal images. And from these two flat images, your brain computes distance and depth using a number of cues. You use relative motion, so that things that are nearer move more than things that are farther away, especially when you move. You use shading, which of course artists can convey, perspective, occlusion, what's in front of something else, and stereopsis, which I'll talk about at the end. You can pay a lot of money for a ray tracing program if you're into computer graphics. Sometimes I come talk to computer graphics people. These are very expensive programs. They compute how light would obey the laws of physics and bounce off various surface angles and textures and so on. But your brain doesn't go around making sure the laws of physics are being obeyed. It's a given. You can assume the laws of physics are being obeyed. And artists have been ignoring the laws of physics for centuries. Here's a painting that's got shadows going in orthogonal directions. Here's a vase of flowers casting a shadow towards, presumably, the source of light. This is Canaletto's Piazza San Marco. And in this painting, which is at one of the Harvard museums, the shadows on this side of the piazza are going left, and the shadows on this side of the piazza are going right. So I was pointing this out to a bunch of Harvard undergraduates, and a docent came over and said, well, that's because the light is being reflected off this building and causing the shadows to. So I went to the Piazza San Marco. There is no time of day when the shadows go in two different directions, but nobody cares. Here's a manger that you seem to be seeing from above on the left and from below on the right. And here's a bunch of, so you wouldn't think that halos would have to obey the laws of physics, but you'd think they'd be at least consistent in whether or not they occlude things or not. So if you can find a painting of somebody looking at themselves in a mirror, I guarantee you it's not obeying the laws of physics. If this lady were looking at herself in the mirror, you, the viewer, would not see her face, right? This is universal, this trope of people looking at themselves in mirrors and the viewer seeing their face. Cross-cultural. So I gave this talk in Saudi Arabia and they made me take all the nudes out. And when I was putting the nudes back in my talk, I realized that they were all female, and I felt bad about that because, you know, it's kind of sexist. But it turns out it's very difficult to find a work of art of a male looking at himself in a mirror, let alone a naked one. But I did find this photograph of Andy Warhol supposedly looking at himself in a three-way mirror. So I rigged up a three-way mirror in the lab and arranged it so that the viewer, the person taking the photograph, could see three views of my face. But what I saw in the mirror was the guy taking the photograph. So here's this painting by Vermeer, and there's a shiny brass bowl, and it looks like the bowl is shiny because it seems to be reflecting the tablecloth underneath the bowl. But if you look closely, you will see that what it's reflecting has nothing to do with what the tablecloth is doing right there. Nobody cares. So another thing that artists figured out, again, way before us neuroscientists, is that color and luminance do different things for you. So Picasso said, don't worry, I'm going to define luminance and color, okay? Picasso said color is only symbols, reality is to be found in luminance alone. So we have three types of cone photoreceptors that absorb light in different parts of the visible spectrum: short, middle, and long wavelength parts of the spectrum. We see luminance by just summing the input in the three cone types. We see color when the cone types act in opponency, when they're computed in opponency. So in this image, this is the color version of this image. This is the luminance version of this image. Most color contrasts have a luminance contrast, but you can have color contrasts without a luminance contrast. And when you do, peculiar things happen. And artists know that and often take advantage of it. This is Monet's Impression Sunrise. I don't know how it's doing on your monitor, but on my monitor, and actually in the original, go to Paris, it's worth it. It's at the Marmottan. The sun is hot and cold and shimmery and kind of creepy looking. This is the painting after which the Impressionist movement got its name. It's called Impression Sunrise. So I got a small grant from Harvard to go to Paris and measure the luminance in the original at the Marmottan. And it turns out that despite the fact that the sun looks absolutely brilliant in this painting, it is actually exactly the same luminance as the gray cloud background. And I think it's because there's no luminance contrast that the sun has this hot, dark, hot, cold, light, dark, shimmery quality. And that is because of the way your visual system handles color and luminance information. So light pattern hits the retina, goes through the lateral geniculate nucleus up to primary visual cortex, but then it splits into these two major processing streams that do quite different things for you. And we know that they do different things because of the deficits that people can get when they have lesions in these two different parts of their visual system. So patients who have lesions in the ventral or temporal stream have difficulty in recognizing objects. So we sometimes call this the what system. What is it, right? You get what are called agnosias, inability to recognize objects even though you can see them. This part of our visual system is evolutionarily new. It's a primate add-on to the basic mammalian visual system, which is represented more by our dorsal stream, which seems to carry information about motion, depth perception. The cells there carry information about motion or depth or spatial organization. So patients who have lesions in this part of their visual system have problems with spatial organization. So these patients were asked to draw, these patients with parietal lobe lesions we're asked to draw a bicycle, a cross, and a clock. So you can see that they've got all the bits right with each of these kinds of information, but the bits are in the wrong spatial organization. So that's why we call it the where system, but you can also call it the how system because it may be how we physically interact with our environment. But what's important for what I want to talk about now is the fact that this evolutionarily old part of our visual system is, like other mammals, essentially colorblind. And I said mammals, okay? Fish, birds, and insects have color vision, but among mammals, mostly only primates have good color vision. So if you put a red spot on a green background and take a black and white photograph of it, you're going to get a gray spot on a gray background. And you could imagine tailoring the brightness of the red in such a way that you got the same gray for the red and the green. That's equal luminance, and weird things happen at equal luminance that has nothing to do with the saturation of the color. So for me, on my monitor, this particular spot has that shimmery, hot, cold quality, and this one has that. I don't know about your monitor. So I want to try to convince you that your ability to see motion, depth, spatial organization, and figure ground segregation are carried by a colorblind part of your visual system. Oh, and I can't do this. Never mind. All right. So if you look at this image, you can see that the artist has gone to a lot of trouble to convey information about depth from perspective and shape from shading. So what I normally do when I actually go places to give talks, but I'm not going to do that anymore. I love Zoom. Is I put a piece of red filter paper in front of the projector and get rid of the blue because blue doesn't pass through a red filter. So instead, what I'm going to do now is just turn the blue black. So I'm taking information away from this image. Yet I hope it looks more three-dimensional when I introduce a luminance contrast. And now the shape, the shading and the perspective are giving you a sense of three-dimensionality. And in this image, again, it's kind of shimmery, right? And it's hard to see the depth from perspective. It's even hard to do figure ground segregation and figure out what's part of what. But when I take away the blue, remove information from this image, I hope it looks more three-dimensional because it's a better stimulus to your colorblind depth system. Monet painted a whole bunch of versions of this cathedral in Rouen, and they're all about how light bounces off the deep rose window and the doorway. And all the information about the shape is there in the luminance version. But in some of these versions of this cathedral, it looks flat and shimmery. The building faces west, and this is the Rouen Cathedral at sunset. And it does look flat and shimmery, and Monet achieves this sensation by using colors that are just about equal luminant. And this is a print by Lichtenstein that I found in a book, and it's a part of a series called the Rouen Cathedral, and it doesn't even look like the Rouen Cathedral until you look at it through a red filter, getting rid of the blue, and then you can see the cathedral. Andy Warhol loves to play with equal luminance. This is the bottom half of a print of the Brooklyn Bridge where the two colors are approximately equal luminant. And I hope the luminance contrast version of the same thing looks more three-dimensional. And his big electric chair series, again, it looks kind of flat and weird and shimmery, and the luminance version looks much more three-dimensional, and you can even figure out what it is. So if your ability to see depth from various cues is carried by a colorblind part of your visual system, it shouldn't matter what colors you use as long as the luminance is appropriate. And of course, artists know that. This is Andre Deuran's portrait of Matisse, and the shadows, the planes of his face are all weird colors, right? And yet, if you knew Matisse, you'd recognize him right away from the shape of his face because Durant has used exactly the right luminance despite the weird colors for you to recognize Matisse. And of course, Matisse, in his famous Lady in a Hat, which started off the Fauvist movement and everybody hated it when it first came out, what he was doing is exactly what Picasso said, which is using color symbolically, but the reality, that is the shape of this lady's face, is there in the luminance alone. And sometimes artists say value does all the work. Oh, I forgot to say this. Value is the word artists use for luminance. It means exactly the same thing. Value or luminance does all the work, but color gets all the credit. So now I want to try to, so I hope I've convinced you that your ability to see depth, spatial organization, and figure ground segregation are carried by a colorblind part of your visual system. Now I want to try to convince you that your ability to see motion is also colorblind. So in this demonstration, these two rectangles are moving at exactly the same speed, but when they alternate, whether they're in front of the light or the dark stripes, they seem to slow down. So when the yellow is in front of white, it's a suboptimal motion stimulus. When the blue is in front of the black, it's a suboptimal motion stimulus. So that's how dependent on luminance, not color, is your motion system. If your ability to see motion is colorblind, then your ability to see that something is not moving is also colorblind. So Monet is famous for achieving a sense of motion in his paintings. I don't know that he ever said he did this in any particular way, but he often used equal luminant colors. And so here the poppies are almost exactly the same luminance as the green grass. And so your what system can see them, but your where system can't. And so they can seem to move in the breeze. His water, he's famous for his water moving, and I think he achieves this by using different colors that are all just about equal luminant. So Monet didn't talk about doing this, but I know that Mondrian knew in this painting that he'd achieved a sense of motion because the title of this painting is Broadway Boogie Woogie. And it's the originals at the MoMA in New York. It's huge, and it's almost nauseating the way it jumps around. And I think it jumps around because the yellow checks are just about equal luminant with the off-white background. Advertisers like to do this. If you open a Wired magazine, you'll doubtless find an ad or a headline with text that's the same luminance as its background, and it jumps around and it's jittery. And I think advertisers do this because the jittery quality is attention getting, but it's also hard to read, which is a goal, I think, of advertisers, not to get you not to read it, but to read it more slowly. And I'll get to that at the end. So I hope I've convinced you that your ability to see motion, depth, spatial organization, and figure ground segregation are both colorblind and computed locally. Now I want to try to convince you that your ability to see color is low resolution. So of course, artists know this, probably knew it way before us neuroscientists. Pastel artists and watercolor artists will often have an outline and a color that is not they don't color inside the lines, and yet you assign the color to the outline. Here's an illusion based on that. I hope you see the donut as yellower than either the whole or the background. And I see it too, right? But it's not. This donut is exactly the white of the back of the whole and the background. This is just a yellow line inside a purple line. But you see this yellow spreading. Your visual system allows the color to spread because it's low resolution until it hits a luminance contrast border. And now I want to use after images. Again, you're going to run out of transmitter or something in order to show you blurry colors filling an outline. So what I need you to do is look at the intersection of these three shapes, and then I'm going to show you a homogeneous gray field, and you'll see a little color after image, right? Now, I want you to look at the intersection and see if you don't see a better after image when there's an outline to constrain the colors. Now, some of you are saying, you're sophisticated, that's just because you did it twice. No, it's not it. look at the intersection, evaluate the after image, look at the intersection, evaluate the after. It's really better when there's an outline, right? So now look at the intersection and see if you can get that color to fill a completely different shape. And look at the intersection and watch the colors squirt out the hole. So here's another work of art that relies on resolution. So this is a very famous work of art, right? It's the Mona Lisa. It's at the Louvre. You can't even get near it unless you go at 7 o'clock in the morning. I was writing this book about vision and art, and my editor said, it's obvious I know a lot about vision. I do, but it's equally obvious I know nothing about art history, which is unfortunately also true. So he had me read this art history book. So I got hold of Gombrich, great art history book, but I only got to the Renaissance because I got to this part where Gombrich says, there's a good reason why this is such a widely loved painting. The Mona Lisa looks alive. As you look at this painting, her expression changes. It's dynamic. So I looked at it, and I'm going to make it big for you to do this. What I want you to do is look at her eyes and think about how much she's smiling. And then look directly at her mouth and think about how much she's smiling. Look back and forth between her eyes and her mouth and see if you don't find that she seems to smile more when you're looking at her eyes compared to when you're looking at her mouth. So Gombrich says this is because Leonardo used sfumato, which means he blurred it, right? But if it depends on where you're looking, that's not having to do with your mental attitude or ambiguity or anything. That's really low level visual stuff, right? It has to do with the fact that your acuity falls off with eccentricity. If you're looking at this central dot, all of these letters are equally readable. That's how much your acuity falls off with eccentricity. And so if you filter the Mona Lisa in such a way that you'd see what she'd look like to your peripheral vision, compared to what she'd look like if you looked at her with your central vision, if you could see the whole thing with your central vision all at once, which of course you can't. She's smiling much more in the low spatial frequencies, in the blurry parts of the image, when you're seeing her mouth with your peripheral vision. So as you move your eyes around the painting, which you have to do, you have to move your eyes around constantly or your vision fades. You move your eyes 3 times a second. So it it gives a dynamic quality because of your eye movements to a static image. And 500 years ago, a dynamic quality was something quite special. And I think it also gives her a coy quality. So you're looking at the background and you notice that she's smiling and you quick try to look at her mouth and she stops. And here's something that Nova did. I hope this works, that gets at it. I think the same thing happens with post-impressionist pointillist paintings. So people carry on about the wonderful, brilliant quality of the colors in pointillist paintings. The pointillists themselves thought that the brilliant quality of their colors came from the fact that you were using additive color mixing. That is, the color was in separate dots and you were combining the reflectances, not the absorbances of the pigments. But magazine illustrations are made up of little dots of color, and there's nothing special about the color in magazine illustrations. I think the jazzy, brilliant quality of the colorless paintings comes because your central vision sees the dots, your peripheral vision merges the dots into boats and trees and so on. And so as you move your eyes around, it changes from dots to rivers. And I think it's this change, this dynamic quality that's brilliant, not the additive versus subtractive color mixing. Chuck Close goes even further. He made these giant tiles of color. So your central vision sees the tiles, your peripheral vision sees Chuck Close. and photo mosaics. Your central vision here sees baseball cards, your peripheral vision sees Babe Ruth. So now I want to talk about even higher level vision. I've been talking about pretty low level visual stuff right now. Now I want to get into your what system and ask, how do we recognize objects? So we're primates. We're very good at recognizing faces. You're recognizing faces and particular individuals in a tenth of a second. And you can do this whether they're degraded or cartoons or occluded, and you can do this better than any computer program. Again, artists are way ahead of us neuroscientists in figuring out what might be key in how we recognize individuals. Artists have known for millennia that a caricature conveys an individual, a personality, better than a veridical line drawing. Why should a caricature, which looks less like that individual, convey so much more about that individual than a veridical line drawing? Well, we study monkeys, and monkeys, like humans, recognize each other by their faces and use them to convey important social information. And like humans, monkeys have specialized regions in their ventral or what system that are dedicated to processing faces. And we can record from these neurons in monkeys to figure out what it is about these neurons that encodes our ability to recognize faces. So if you record from these face selective neurons, they respond to faces. They respond to human faces, monkey faces, cartoon faces, anything you'd call a face. And so we use the ability of these neurons to respond to cartoon faces to figure out what about faces they encode. So we parameterized a bunch of cartoon faces, changing the eyes, the shape of the face, the hair, everything we could think of. And we presented these variations on each variable simultaneously in this kind of an image. We're varying everything pretty far out in each of these parameters. And we measured the firing of a bunch of face cells in response to this continuously changing face stimulus. And here's the only science you're going to get in this talk. Each row is one face cell. Each column is one variable, one parameter. And you can see that each of these three face cells shows tuning, that is selective responsiveness to just a handful of these variables, but each cell shows responsiveness to a different handful of variables. So you can imagine that if you had a population of face cells, you'd pretty much cover all these different variables and you could encode faces. So you might imagine if you had cells that cared about inter-eye distance, for example, and these two cells care about inter-eye distance, that you might want to encode inter-eye distance by having a bunch of neurons, each of which gave a peak response at a different plausible primate face inter-eye distance. Or you could just have two cells, one that responded best to big inter-eye distances and was inhibited by small, and an opponent cell that was activated by small inter-eye distances and inhibited by big. This is a much more efficient coding scheme because you only need two neurons and you get every variable in every value in between from the ratio of these two. And that is indeed what we found, ramp-shaped or extreme tuning to pretty much every parameter we looked at. So ramp-shaped or tuning to extremes means that face cells encode how the face you're looking at differs from the average face. And that explains the power of caricature, right? What is caricature? You take a face, you figure out what's unique about that face, how it differs from the average, and then you exaggerate it. So essentially, face cells are taking a caricature of the face they're looking at. And of course, artists know this. And here are some portraits by Picasso of a bunch of different people. And of course, they look weird because they're by Picasso, but I've put each portrait next to a photograph of the person they're a portrait of. This is Igor Stravinsky, Gertrude Stein, Picasso himself, some guy he roomed with, wife, girlfriend, wife of a friend, mother-in-law, art dealer. In each case, he's figured out what's unique about each of these individuals and exaggerated it or captured it. So ramp-shaped extreme tuning means that face processing involves within category comparisons because you can't say how a face differs from the average unless you're comparing it to the average. And it also means that face processing, like all those other low-level visual tasks I talked about at the beginning, is also local and opponent. What do I mean by that? When you march across early visual areas, you march across visual space. That is, it's retinotopic. When you march across higher level cortex, temporal lobe, you march across categories. So if you're recording from one part of inferotemporal cortex, you'll be finding face cells, and you move over a bit and you'll find body cells, and you move over a little bit more and you'll find scene cells. So they're clustered by category. So that means that within category comparison means local opponency, which also means that you ought to get after images. So what I want you to do now is look at these little numbers while I natter on about this. What I want to show you is that you can get an after image in face identity, just like you can get an after image from a white spot. And the reason I'm having you look at these little numbers is because I don't want you to get a stupid little low-level retinal after image. I want you to get a high-level face identity after image. So now I'm going to have you look at two identical morphs, and I hope you get a face identity after image. So that means that face processing, just like luminance and color and motion, you have excitation by some features and opponency or normalization by others. So in order to get normalization or opponency, you have to have this extra inhibitory neuron, which takes a little time, a few milliseconds. And you can see the dynamic quality of this interaction by these little black, they're illusory, these little dark spots that are everywhere where you're not looking, as the surround opponency kicks in. So here's so opponency is local, causes local gives you local contrast in space, but also in time, because it kicks in after the initial activation. So this is a recording from some sensory neuron. I don't even know what kind of sensory neuron it is. It could be visual, it could be auditory, it could be somatosensory. The stimulus turns on, the cell fires like crazy, and then as the opponency kicks in, the cell stops firing as vigorously. That's why you need to move your eyes around, because vision fades with time as the opponency kicks in. And you can see your eye movements in this image made by Aki Kitaoka. And if you like this, I highly recommend going to Kitaoka's webpage, Kitakoa. You move your eyes constantly in order to see and in order to scan the scene with your high resolution fovea. So your visual system maintains, so as you move your eyes around, the whole visual image on your retina is constantly changing, and yet the world looks stable. This is an ongoing mystery to us vision scientists. Your visual system maintains constancy across saccades by suppressing vision during saccades and by discounting changes that occur due to and therefore during saccades. We don't understand how we do this. I think film editors possibly have more insight into this than us neuroscientists. So if you look at this cross really hard in the center of this screen, you can get that big purple spot in your upper left visual field to fade away. And as soon as you move your eyes again, the purple spot will reappear. That's fading. If you look fixedly at the B at the bottom, you can get this whole thing to fade to blue. And then if you move your eyes up to the top, it becomes a split color again, and then you can get it to fade to pink. That's called Troxler fading. It has to do with the fact that you need to have change in order to see anything. You can also get it in snowstorms, which is called, or sandstorms, a whiteout. And I think it's not unrelated to the mystical quality that everyone attributes to Rothko paintings. He makes these big, blurry paintings that you have to stand really close to in museums because they make you stand close to them. And they tend to fade. And it may seem creepy and mystical, but I think it really is just Troxler fading. So you look out on the world and the world looks stable. So close one eye and tap the lid of the other eye. That's how much your vision should jump around because you're moving your eyes. Yet it doesn't. Oh, I said to do that. So film editors have figured something out because they have the same problem with cameras. When they want to move their camera to another scene or to another part of whatever it is they're filming, they know to cut on motion. That's a rule in film editing, cut on motion. And here's a demo of how motion can suppress change. So those dots were changing color constantly. They're still changing color constantly. But when this thing moves, you stop seeing the change. And here's another demo of the same thing. Oh, whoops, I missed one. Come on. I want to go back one. Ha, it's just missing. Well, never mind. Oh, here it is. So the shapes are changing, right? Dramatically. And yet, when the thing starts moving, if you look at the center, don't look at the things, you don't see the change, right? The change disappears when you've got motion, cutting on motion. So here, I made this one. The shapes are changing dramatically, right? But if you track the little smiley face at the bottom, you don't see the change. And as soon as you stop tracking, you see the change again. And here's something I downloaded. So some neuroscientists think that you remap the visual world according to where you're moving your eyes. But I'm with the film editors. I think you just ignore the changes that happen when you move when anything moves. So I hope I've convinced you vision is information processing, not image transmission. You can see this in the artwork of many artists. You can even see this in the artwork of children. If you ask a child to draw a person, you will get a face on legs. Any 3-year-old child. They're telling you that the information about somebody's identity is there in the face predominantly. And so that's what they're giving back to you. They're giving back to you the information they've extracted in an information rich format. Here's a drawing a child made of her pregnant mother. A face on legs inside a face on legs. And Matisse says this explicitly. He doesn't paint things, he paints the differences between things. That's where the information is. I once got invited to a panel discussion on dioramas. Those are those things you put in, school kids here put them in shoe boxes, you know, you do for social studies. Anyway, they were exquisite. And the artist kept saying they were trying to get the viewer to see things differently. And I kept puzzling about this. How do they mean they want you to see things differently? These were scenes that were maybe boring or maybe slightly creepy, everyday, mundane things, and yet they were exquisite. And the artist kept saying, we're trying to get the viewer to see things differently. And I had to puzzle about this for about a month before it finally hit me what they were saying, or at least how it relates to how I understand how the brain processes information. The temporal lobe, our what system, is divided up into all these category selective domains. And we know this from the different kinds of agnosias that people can get. You can selectively lose the ability to recognize faces, places, text, bodies, or manipulable objects or tools. So that means that your temporal lobe must be divided up into domains that selectively process these different object categories. And we know this now from fMRI that we do have clusters of neurons that process each of these different categories. And we're expert at processing these different categories. So having a domain probably allows you to be expert at processing these things because you have nearby neurons encoding the same category, which means that you can make fine within category distinctions. So you may not think you're much of an expert at processing faces, but here you are not using your expert face cortex because this is not how you typically learn to see faces. This face is upside down. Typically you see faces upright. You know that this is the Margaret Thatcher illusion. This is the Margaret Thatcher, but you know that I've manipulated the features within the face. But you're not using your expert cortex. You're using your non-face cortex to process this. When I turn it right side up, now you're using your expert cortex and it looks different, right? And every time I see this, I'm always astonished by it. So you are an expert at processing faces. You just don't think you are. And you learn to see things in the way that you typically experience them. So presenting something at a different size than you normally experience, it gets you to see it literally with a different part of your brain. So I think that's why artists like to manipulate scale and why you do literally, as the artist kept saying, see it differently when it's at a different scale. And of course, artists have known this for a very long time. And sometimes just putting something in a different place gets you to see it differently. Again, you're now seeing this with your non-expert, your non-face cortex, and now you're seeing it with your expert cortex. So I could stop here to keep your symposium on schedule, or I could spend 10 minutes talking about learning disabilities and stereopsis. It doesn't matter to me. You tell me what I should do because I don't want to get your symposium off schedule. I can't hear anybody. Uh-oh, I know what I need to do. I need to turn on my audio to be able to hear you, and I have no idea how to do this.
Can you hear me?
Wait a second. Ah, there you are. There we are.
Can you can hear me now, right?
Yes, but not very well. Let me turn up my sound here. Yes. What? Do you want me to stop now? You have to tell me.
So so it's 6 o'clock here, local time, for your talk. But I guess your talk is so engaging that if you want to continue for 10 more minutes, we can do that, and then we're gonna have a very short time for questions and answers.
That's good. I love that. Sorry. So stereopsis. You have to have red-green glasses. So go find yourself some red-green glasses someday and Google anaglyphs and have fun looking at three-dimensionality because of your ability to encode stereopsis. So, and magic eye images for those of you who can cross-fuse. Don't worry about it. I'll tell you about it. If you're looking at an object in a particular depth plane at a particular distance from you, that object casts images on corresponding parts of your two retinas. But objects that are closer or farther away than what you're fixating on cast images on non-corresponding parts of your retina. And those teeny weeny differences in the two images on your two retinas allow you to see are part of your computation of distance and depth. If you can't do it, don't worry, 10% of otherwise normal people have no stereopsis. But if you go to a museum and you see a painting that's got a lot of shading and perspective and depth, your stereo system is saying, no, sorry, it's flat. If you want to see more depth in a work of art, cover one eye and it will look more three-dimensional because your stereo system is no longer telling you that it's flat. But it's a difficult, computationally, it's a difficult problem. If you had red-green glasses, you could suddenly see an image in this thing that you can't see at all without red-green glasses. If you have 4 identical images in the real world, the corresponding images on the 2 retinas correspond to this reconstruction, but if you mismatch the images in your 2 eyes, they correspond to the same object, just at a different distance or depth. And the Impressionists and Post-Impressionists talked about painting the air. I think they did that because you can have all these little speckles that you misfuse in your 2 eyes, and that corresponds to a volume of speckly stuff. And this painting looks unpleasantly three-dimensional to me because my eyes misfuse the different leaves in the painting. But what's fun about this is it was painted by a guy with no stereopsis. How do I know that? This is a photograph of Klimt, who painted that painting. He is cross-eyed. He could not possibly have had stereopsis, because in order to have stereopsis, there are very small differences in the 2 retinal images. So your eyes have to be extremely well aligned in order to compute distance from these tiny differences in the 2 images. And I got interested in this because of something I'd read about dyslexics, which is that black and white text for them looks shimmery and kind of slides around on the page and looks hot and cold and unpleasant, which is exactly what equiluminant text looks like to me. And so I asked myself, well, what is it about the dyslexic brain that might make them see things the same way I see equiluminant images? And so we measured the visually evoked potentials in a bunch of dyslexics and control subjects and discovered that there was a slight slowing in the visually evoked potential at low contrast and fast speeds. And that VEP is characteristic, oddly, of the dorsal stream, not the ventral stream, which is important for motion, depth, spatial organization, and figure ground segregation, not text, which is carried by the ventral stream. So why should fluent reading depend on the dorsal stream when text recognition is carried by the ventral stream? Well, there are other symptoms in dyslexia besides a selective problem learning to read, despite normal education and normal intelligence. And people with dyslexia often have figure ground problems and trouble gauging distance and depth, both of which are carried by the dorsal stream. They also have auditory differences, which may represent a dual modality in the auditory stream. And it may explain why. So when I was working with this in this study, I was privileged to meet a number of people with dyslexia, and it was astonishing how many of them were incredibly talented artists, musicians, actors, and computer programmers. It was inescapable. And the usual explanation, so our dyslexics are overrepresented in the artistic population, but the usual explanation is because they were no good at academic subjects, so they spent all their time in the art room. But these people were so talented, I decided to think about, well, maybe there's something about the dyslexic brain that makes them better at music and art. So the difference in the auditory system may explain the prevalence of dyslexia in musicians, but what about artists? Why should artists be dyslexic? Well, how about trouble gauging distance and depth? So if I try to draw a chair, I'm like the child drawing a face with legs. I have extracted information and I am now stuck with that information and can't get back to the flat retinal image that I originally had, which is what you need to do in order to make a realistic rendition of it. So maybe a kid who sees the world as flat has less trouble flattening the world in order to draw it. So you can tell if somebody could have normal stereopsis by looking at the light reflex in their 2 eyes. Whoops, can't do that in this one. So Chuck Close, he was a famous artist. He had anisometropia. He saw the world as flat and he was severely dyslexic. So you can tell if somebody's eyes are lined up and they could have normal stereopsis So 10% of otherwise normal people do not have stereopsis if the light reflex is in the same place in their two eyes. So can you see the light reflex in everybody's eyes? And you can see that they're always in the same place. Are you good at this now? It's a legitimate ophthalmological test, the Hirschberg test, to test whether eyes are aligned from a photograph. There is one baseball player who had no stereopsis. This is Babe Ruth. Light reflex is centered in this eye, off center in this eye. He had amblyopia, that is, he was probably misaligned eyes when he was a kid and suppressed the input to the brain from the deviant eye, which I suspect is this one. So only 3% of the population has significantly misaligned eyes, even though 10% of the population has no stereopsis. Only 3% of the population has no stereopsis because their eyes are misaligned. But you can diagnose this from a photograph, which is key. We went to, I think it was Northeastern University, and measured a bunch of business school students who had better stereopsis than a bunch of art students, and their eyes were worse aligned in the art student population. But it's more fun. We don't know if these art students would have become famous artists, but it's more fun to look at famous artists. So this is Andrew Wyeth. Every photograph I've ever found of him, one eye is looking straight ahead, the other eye is looking off to the side. His father, N.C. Wyeth, was an illustrator, and he also is strabismic. I am very sad to report that Jamie Wyeth, Andrew Wyeth's son, is not strabismic. Bummer. Edward Hopper, light reflexes near here in that eye, off to the side in this eye. Kara Walker, the light reflex is always in a different place in her two eyes. Kara Walker works in silhouettes. How flat can he get? I don't know about the dog. Marc Chagall was cross-eyed. Frank Stella was cross-eyed. Jasper Johns was wall-eyed, his eyes deviated out. Robert Rauschenberg was wall-eyed and severely dyslexic. I read an article a few weeks ago about Anish Kapoor, who said he was dyslexic, so I found some photographs, and indeed his eyes are misaligned. Alexander Calder, his eyes are misaligned. Sam Gillam. I don't know if Andy Warhol he doesn't read. And I read an article recently that said that Kanye West doesn't read, and sure enough, his eyes are misaligned. Man Ray, Roy Lichtenstein, Egon Schiele, Diego Rivera. So it is a legitimate ophthalmological test to diagnose strabismus from a photograph. Obviously, it's not a legitimate test to diagnose it from a painting, but I was at the Louvre. There's one tiny room with one Rembrandt self-portrait on each of the four walls, and in all four of them, one eye was looking straight ahead, the other eye was looking off to the side. And I know when you paint a self-portrait, you look in the mirror, you paint the right eye, you look to the right, you paint the left eye, you look to the left. But here's a montage of me looking at my right eye, looking at my left eye. And indeed, the light reflex is in two slightly different places. But my eyes aren't, and I do have stereopsis, my eyes are not nearly as deviant as Rembrandt portrays himself. So a young colleague came into my office. I had this pile of pictures of Rembrandt self-portraits, and he said, "Is it always the same eye?" And I said, "No, sometimes it's the eye on this side that deviates, and sometimes it's the eye on this side that deviates." And my young colleague, who was himself a stereo blind artist, said, "Marge, you have to separate the paintings and the etchings." So when we did in the paint, and we made a little program where we could measure where the iris was inside the eyeball. In the paintings, it was always the eye on this side that deviates. In the etchings, it's always the eye on this side. When you make an etching, you scratch it on a plate and then you flip it over, right? So when we did that, we discovered that in the paintings, it was always the eye on the right that looked ahead, the eye on the left that was deviant, except for this one, which is in Berlin. So it's either a fake or he painted it from an etching. And in the etchings, it's the reverse. But you do not have to poke your kid's eye out in order to get him to be a good artist. The first thing you learn in art school, close one eye, look in a mirror, turn it upside down in order to flatten the image. And here are my take-home messages. Important one, vision is information processing. There are different parts of your visual system that do different things. You don't have to color inside the lines. And people, this one's important too. People are different. And something that may be a handicap in one job may be an asset in another. And now I'm done. And I'm happy to take questions. What am I supposed to do now? Because I can't hear you. I've turned something off.
Can you hear us now?
Oh, I know. I've turned this off. There we go.
Marge, can you hear?
Yep, I can.
Can you hear us?
Yes. Yes. But
Hello?
I know somehow you've turned off my audio.
I
don't know
know
how.
I think there is a problem with their
Ha!
side. Yeah.
Whew, I didn't do it. I didn't do it.
Can she hear us?
Yes, I can.
Okay. Thank you so much for the very interesting and engaging talk. I guess we have a few minutes for a few questions before the next talk. The people in the room, if you want to ask questions, I can you can give the microphone. Also, can you show the room to Marge? Can you show so she can see people?
Uh-oh.
Hello, and thank you very much for your very exciting and insightful speech. Can you hear me, Professor?
Yes.
Thank you. I was very grateful to hear your speech. Thank you very much. And I was wondering about the notion that you were talking about, that artists were almost always ahead of neuroscientists in understanding the neuroscience of viewing the world, as I understood you, if I was right. But was there any example of wrong perception of artists of the neuroscience of the world view, neuroscience of the light perception?
Whoops,
Was I clear?
not the very last part.
I was asking if you were talking about the similarities and the correctness of perception of artists from light perception and the neuroscience of this that is discovered very soon, that discovered very recently. But was there any misconception about light perception by the artists that you have noticed?
Oh, the only one is the post-impressionists who think that they were using additive color mixing. They were convinced it was additive versus subtractive color mixing.
Oh.
But otherwise artists, they don't talk about the neuroscience, they talk about the phenomenology. And of course they're correct about the phenomenology, but maybe not the explanation.
Of course, yes, thanks. And for another question, I'd like to ask you if there is any difference in neuroscience of light perception between artists and lay people, or even different cultures of artists, for example, impressionists, or I don't know, cubism, or anything like that. Are there any differences that have been noticed in the science, in the neuroscience, between them and lay people?
Artists have learned to see things. So for example, one of the phenomenology is that if you look at a corner of a room that's painted white with a white ceiling, a layperson will look at the corner and see the same white, whereas an artist will see that whatever wall is shaded is a different white from the rest of it. But it's simply learning to notice stuff rather than seeing differently, I think.
Yeah, thank you very much. I don't have no more questions.
All done?
Thank you for your great lecture. That's one of the greatest lecture I've ever seen in my life. I just wondering, as you told in this lecture, artists are always ahead of neuroscientists. But I just have a question. In your opinion, these artists do this on purpose? I mean, this kind of uh, illusions that we have see here, are artists are aware of these things, or they just do it on their unconscious? Are they do it consciously or unconscious? I mean, are they aware of doing this kind of stuff, or they're not aware?
So I go to art schools, and artists experiment. They do stuff. They see if it works. I think they are quite conscious, not of the underlying neurobiology necessarily, but of the phenomenology. There's a book full of Matisse's drawings that he made, and you can see over months and months and months that he's abstracting these distortions that he uses, and each time, if it works, if it looks right to him, he goes a little further with it. And so it's really a series of experiments. I even talked to Calvin Klein, who makes blue jeans, right? And he says he tries something and he asks himself if he feels it, and if he feels it, he goes in that direction. If he doesn't feel it, he tries something else. So yes, they do experiments just like we do.